E no seu doutoramento, o Christos fez um trabalho sobre a ecologia e biologia do Economidictis pigmeus no Lago Pamavotis, em que teve a estudar várias espécies da biologia deste pequeno góbio, e um dos capítulos foi avaliar as relações filogenéticas e em relação com a geografia da zona do Mediterrâneo Oeste, do grupo dos Sun Gobies. E agora vamos passar em inglês, por isso Christos is going to talk about the Sun Gobies, the original Sun Gobies in Greece. Okay?
Good afternoon. As Philip said, I'm going to speak about the Sun Gobies from Greece. In my PhD, mostly I studied the behavior and ecology of Economidictis pygmeus, but also the second part, I studied the phylogenetic relationships of Sun Gobies doing traditional morphometrics and the mitochondrial gene CO1 to see the patterns between them. So the basic lessons that are going to be addressed today, it's the history of Sun Gobies, how it did they speciate, and what were the circumstances in the past which promoted the differentiation of the various species involved. Gobies belong to the suborder Gobiidae. There are approximately 2,000 species, and yet there are new species that are still discovered. They have different types of ecosystems. You can find them in freshwater, brackish, and marine environments. And generally, they show a wide adaptive radiation. As you can see here in the picture, there are gobies with different sizes and shapes. I think it's worth to mention the mudskippers, which have adaptive and amphibious lifestyle, which is separate behavior from the other gobies. And now some general characteristics about the gobies. They are characterized by a low dispersal, and because of that, these two guys make a comparison with the hobbits. That if you remember, hobbits usually don't go out of their home. They don't go out from their home easily. They are small body size. It's a group of fishes that contains fish with less than 10 centimeters. They don't have distinct pelvic fins, but instead the pelvic fins are fused and create a ventral disc. They have piriform legs, which are attached in the substrate and with filaments and permit better water circulation when the male is found in the eggs. They also, during the reproductive season, show courtship behavior. The male courts the females and they produce sounds. I have here a video where you can see it's the species Lipovitsa puctatissima. We saw the video in Italy. The male is leading the female in the nest. If we have the sound, you could hear the sounds. It's not, the volume is on, it's not on.
No.
The male is producing sounds and the female is depositing the eggs by doing this movement, the upside movement. And later, it's not in the video, the male is doing the same movement and fertilizes the eggs. In the Mediterranean Sea, there are approximately 50 freshwater goby species that belong to 18 genera. With the exception of Patagobius species, there are two evolutionary lineages. The Parathethian gobies and the Sun gobies. Parathethian gobies have a Proto-Caspian origin. Million years ago, these two seas were connected and were part of the Tethys Sea. Later, the Tethys Sea divided to Mediterranean Sea and Parathethian Sea, which is the yellow line, and separated the gobies that were there. And later on, the Parathethian Sea divided to what we know today, the Caspian Sea, the Azov Sea, and the Aral Sea. The Sun gobies is a monophyletic group. There are several studies to support that using morphological, osteological, behavioral data. And they have affinities with Atlantic Mediterranean, Proto-Caspian, and West Balkanian fauna. The divergence of some gobies from the rest of the gobies happened during the mid-Miocene. However, inside the group there are several paraphyletic problems due to recent genetic differentiation and to continuous speciation events. During the late Miocene, after the separation of Parathethian Sea from the Mediterranean Sea, we had the closure of the Strait of Gibraltar because of the tectonic activity, so the Mediterranean Sea became a closed sea. It started to dry up, to desiccate, And that resulted in a creation of a huge deserted areas with big olive-aligned and Persian-aligned lakes. These movements create the separation of Caspian Sea from the Mediterranean gobies and also the separation of the ancestral forms of some gobies. Later on, during the Pliocene, we have the refluting with fresh water from the Caspian Sea to the Mediterranean Sea. These movements established brackish conditions to the Mediterranean Sea, and we had the creation of the Lagomare lagoons. These movements allowed the dispersal of the Caspian gobies to the Mediterranean Sea and triggered the speciation of the new gobies with Caspian origin. After the dispersal from the Caspian basin, we had the reopening of the Strait of Gibraltar, so the seawater came into the Mediterranean Sea and eliminated the Lagomare lagoons. This resulted to the survival of Papadoschius canestrini in the North Adriatic Sea and the colonization of fresh water by the other species like the other genus like Lipovicia, Economidictis, and Patagobius. The Sun goby group includes 5 species. In marine environments, you can find the genus Gobiusculus and Madocystus. In estuaries and fresh waters, the genus Lipovicia. Economidictis is a fully freshwater genus, and the Canogobius is endemic to the Caspian Sea. Because the gobies look very similar when you look at them, some researchers have identified several diagnostic approaches to identify the species. These are the anatomy. Gobies used to have slimmer body and longer caudal pedicule relative to the body length. They also show sexual dimorphism. Males have deeper caudal pedicule because during the reproductive season, they dig out the nest and oxygenate the water with the movements of their tail. They have also a head canal lateral line system. Gobies don't have a lateral line system in their body, but extending the head, they have these canal pores and the sensory papilla, which are represented by Greek and Roman letters, and the presence or absence of this canal or sensory papilla can help the examiner to identify which is the species that he's going to look at. We can measure also some meristic characters from first from the number of vertebrae in the abdominal and caudal area, the sequences of the dorsal fin pterygiophore, and the number of articulated caudal fin veins. Another useful character is the squamation of the body. Usually the large gobies have a complete squamation. They are covered with scales from the head to the caudal fin. However, in some gobies there are no scales in the head, and there is a progressive reduction of scales in their body. Another thing also is the nuptial coloration. During the reproductive season, males and females show different color patterns. This can be the black dotches in the first dorsal fin, or the dark stripes across the body, or the mask, especially the females have this mask as we saw in the video when they are mature. Genus Madocystus includes 12 species, and 3 of them can be found in freshwater ecosystems. These are Madocystus marmoratus, which occurs in the Mediterranean Sea, Madocystus minutus, which has a wide distribution in the Northwest Atlantic and in the Mediterranean Sea, and Madocystus canestrini, which is endemic to North Adriatic Sea. Genus Madocystus also includes some other diagnostic traits which can be helpful in his recognition. These are the retention in the head with the retention of single pore lambda, the continuous squamation along the sides of the body, and the number of the fin rays in the dorsal fin, that is 5 or 6. Genus Lipovicia includes 18 species, and according to the salinity tolerance, they are divided in 2 groups. The first group, which are the real species, have full squamation along the body to the caudal fin, and also there is a presence of the head lateral line system. However, the stenoaline gobies, which are mostly endemic gobies from the genus Lipovicia, have a reduction of squamation along the side of the body and a lack or reduction of the head canal pores. And here you can see the distribution mostly of all the species from the genus Lipovicia. Lipovicia caucasia with the black dots has a periphery distribution along the Hellenic Peninsula from the Aegean to the Ionian Sea, and also in Turkey. And the rest of the dolls are mostly endemic Gobiidae species, and their status now is considered, for most of them, highly vulnerable or endangered. Inside the genus Gobiidae, there are some criteria that can help us to the identification. These are the, as I said, the reduction or loss of the head canal pores. The anterior oculoscapular canal that is between the eyes can go up to the snout or before. And also, there is a divergence of these canals before the posterior port kappa, which is before the eyes, in the middle. Genus Gobiidae includes only two species, Gobiidae stigmeus and Gobiidae stichonis. The Gobiidae stigmeus is native to the northwest part of Greece, and Gobiidae stichonis is endemic to Lake Trichonis and is considered the smallest vertebrate organism in Europe. It's the most easiest genus to be recognized. They have the complete absence of the head pores in their head, and also they have this perianal organ, which is unique only to Gobiidae genus. And now we can proceed with the case study about the phylogeny of the Gobi group. As I said in the beginning, there are a few studies about the morphology and the genetic analysis of this specific group. Most of the studies were dealing, in the genetics, were dealing with spermatoschistus species from the West Mediterranean Sea. And for the morphometrics, there were only descriptive studies about the morphology of the species or the osteological structures or the reading of this head canal lateral system. So the objective of this work was to examine the pattern and the extent of morphometrical variation in some gobies population, and to assess the degree of genetic differentiation between the some gobies and relate the results with oceanographic data. We made a quite sampling across the Hellenic Peninsula from different geographical regions, that each region is represented by different color, from the Aegean and the Ionian Sea. And also we included in the analysis some specimens that we got from the North Atlantic Sea, specifically from Venice Lagoon. For the morphological analysis, we collected 269 specimens from 21 aquatic systems. In the lab, we took pictures of the specimens, and in the image, we put them in the image program, and we locate the landmarks in order to create a trace network and measure the distances from 33 morphometric variables. Also, we took some counts from heuristic characters, especially from the two dorsal fins, the anal fin and the pectoral fin. Later, we eliminated the effect of the gallomatic effect using this formula, as you can see, and the multivariate analysis that we used in the study was the principal component analysis, discriminant analysis, and the cluster analysis. For the genetic analysis, we used 87 specimens that we collected from 20 aquatic systems. This is the protocol that we used to isolate and amplify the mitochondrial CO1 gene. For the construction of phylogenetic trees, we used the network joining and maximum likelihood methods. And also we calculated some results from the population genetics only for Gobiidae and Gobiidae because we didn't have enough specimens for spermatoschistus. Considering the meristic characters, we found that there were significant differences for the second dorsal fin, the anal fin and the pectoral fin, where the Gobiidae stigmeus and Spermatoschistus marmoratus show the bigger values. The results from the principal components show to us that there are 6 principal components explain 81.5% of the variability. The first principal component was related to the position and the length of the first dorsal fin and the position of the anal fin. The characters that contributed most to the second principal component were the head part, and the third principal component was related to the size of the caudal peduncle and its distance from the second dorsal fin. The results of the discriminant analysis show a high classification, 91%. As you see in the figure, the first axis separates Spermatoschistus marmoratus, Spermatoschistus canestrini, and Gobiidae panizzae from the populations of Gobiidae caucasica from the Ionian Sea and the endemic Gobiidae nigeri, which occurs in the Ionian era. And the second axis separates economidictis Pinaeus and Primovitia Thessalia from the rest of some gobies. The results from the cluster analysis show that the dendrogram is separated in 3 clades. The first clade includes Primovitia Panitia that clusters with economidictis Pinaeus and Primovitia Thessalia. Apparently here, economidictis Pinaeus is paraphyletic. However, the common characteristic of this clade is that these 3 species are considered the ancestral forms of some gobies, according to the literature. The second clade includes Lipovitia nigeri from the Ionian region that clusters with populations of Lipovitia caucasica from Ionian and Aegean. And the characteristic of this group is that these populations colonized Greece with the glacial events during the Christos. And the third group comprises the populations from North Adriatic Sea from the species Quadroschissus canestrini, Quadroschissus marmoratus. We saw generally from the melithic characters and from the principal component discriminant analysis of the differences in morphometric characters between the species, like the shape of the head, the distance between the 2 dorsal fins and the caudal pentacle. These differences can be related to secondary reproductive traits like the fanning of the caudal fin, the large size of head, and the raising of dorsal fin, because the males usually use these behaviors to identify and approach the female, and for better water circulation when the male is inside the nest. And now some results from the genetic work. As far as concerns the genus Economidictis, we found that there are 8 haplotypes for Economidictis Pinaeus and 1 haplotype for Economidictis trichonis. The most common haplotype was EP1, which was found in 4 populations. The genetic distances for Economidictis Pinaeus were very low, but when we compare Economidictis Pinaeus and Economidictis trichonis, the mean genetic distance was 11.2. We also found, after doing the AMOVA, significant differences for the FCT and FST values between the populations of Economidictis Pinaeus and the 2 geographic areas that this species occurs, Epirus and Tolokanae. And these differences result from the thing that they live in rivers and lakes, which act as barriers and restrict the gene flow between the populations. For the genus Lipovitia, most of the haplotypes were for Lipovitia caucasica, and 20 to 28 of the haplotypes were unique. And here are the difference and the comparisons of the genetic distances of Lipovitia between the genus Lipovitia. However, I want to have your attention here only to the first 2. When we compare Lipovitia caucasica from Aegean and Ionian Sea, we found that there is the highest genetic distance, which was 6.7 compared to the other pairs. And also, when we compare Lipovitia caucasica from Ionian and Lipovitia nigeri that occurs in Ionian region, we found that there was a low distance, 0.5. Also, the AMOVA results show that there were significant differences between the population of Lipovitia caucasica and the 2 areas that were compared, Ionian and Aegean Sea. Probably these differences arising from the fact that there were fluctuations for the temperature and salinity during the ice ages, for the existence of the sea currents and the bathymetric phenomena that occur in the southeastern Mediterranean basin. For genus Quadroschissus, we found there are 8 to 10 haplotypes that were unique, and the average genetic distances were quite high, from 12.6 to 20.4. And the results from the cladogram show to us that the genus Quadroschissus appears as paraphyletic. It comprises 3 separate clades that are highly supportive for the bootstrap values. The position of genus Economidictis in our study can be considered as ambiguous. However, in a recent phylogenetic study, Economidictis Pygmaeus got the basal position. Probably this happened because Economidictis Pygmaeus comes from the Ionian region, which contains many native fish, where in this part of studies usually get the basal position, and they are considered for high endemism rate. Genus Lipovitia in our study appeared as paraphyletic. There are two lineages, the Ionian and the Aegean. And as you can see in the Ionian clay, Lipovica migeri is incorporated to Lipovica kafkasica populations from the Ionian Sea, probably because it's conspecific with Lipovica kafkasica. Also, the other two species, Lipovica phasalia and Lipovica panizzae, are clustered to their own clays. Now, according to some geographic scenarios, probably the dispersal of Lipovica populations to the Ionian Sea were favored by the capsule rivers, the drop of salinity in the Ionian Sea and the fluctuations of sea level that happened during the Pliocene, and the populations of Lipovica kafkasica from the Ionian Sea and from the Aegean Sea colonized Greece during the ice ages of the Pleistocene. And to sum up this segment, we saw that Mediterranean salinity crisis and the climatic conditions during the Pliocene probably shaped the geologic history of the Sand-Gobi group. We highlighted taxonomic uncertainties based on morphological and genetic results in Lipovica and Economidictis. It required further morphological and molecular work to formally describe the populations of Lipovica from the Ionian Sea. The large genetic distances between the Venetian Christos and all the others support the hypothesis of allopathic speciation in the Northern Adriatic Sea. And finally, we saw that we want to highlight the important role of some gobies that can be considered as useful indicators for monitoring the conservation of Mediterranean freshwater ecosystems. Thank you for your attention.
Okay, questions? Sim, verdade.
Christos, you mentioned for one of the species that is endemic to Greece, the presence of a periannual organ.
Mm-hmm.
What is that exactly? Is it like a urogenital papilla?
It's in front of the anal fin, and until now there are some hypotheses why this species has this organ. Some say that they are used to clean the eggs, or they use it to attach to the surface, but they are not sure what is the role of this organ. They are not sure.
More questions? Ricardo.
Então, em inglês, né?
Assim.
I don't know this area very well, but I remember that there has been some work done on freshwater blennies, saladios, around this area, and I was just wondering if the genetic differentiation patterns that you find here, if they're mimicked by the blennies or not, because as you refer, these species don't go very far, and I remember, I don't know Saladia in particular, but I remember that there's a lot of blennies that have fairly long PLDs, so eventually they could disperse better than these.
Very long? Very long, okay?
Clavicular duration,
Okay.
PLDs, so the patterns could potentially be different.
For blends.
For blends.
Freshwater
Like the-
blennies.
So basically your question is what are the patterns of
How do these results compare to the results
patterns
of
of
other
endemicity of the
Same.
Salaria in this
or
area?
other species?
Or other species.
So when you
Mhm.
did this, did you compare this to other species?
No. I compared with some recent results we have from some Belgian guys that use the different molecular markers, 12S and 16S, and we found the same conclusions, that there are separate populations for Lipovica kafkasica. I mean, the two lineages, the Ionian Sea and the Aegean Sea, and the Lipovica migeri probably needs a redescription.
But for instance, Salaria economidictis, is there any genetic study between comparing the Salaria, the river blennies, between Salaria
I think there are, but
economidictis and the other
Salaria trichonica, Salaria
trichonica,
economidictis.
yeah.
But haven't
You haven't idea?
Look at
I think it was the people like Ishba that were involved in doing this.
Yeah, with Ignacio Duarte, yeah, as well. Yeah.
And Vito Romano.
Yeah, okay. Questions? More questions? I have a question. So here you also have this Lipovica migeri that groups in.
Lipovica kafkasica from the Ionian.
So migeri is probably not a valid species,
Yeah.
or is
Even
Kafkas?
it's needs a description and probably is Gobiidae caucasica.
So it's probably the same species.
Mhm.
It was a double description of the same biological entity.
Maybe.
And so you have two different lineages of Caucasica, Gobiidae caucasica, and so in terms of the conservation perspective, which one of these lineages has like a more restricted distribution area? You know, if they are approximately the same, or from the Ionian and the Asian areas?
Is a conservative.
Yeah, in terms of conservation,
Mhm.
if they have a wide distribution, from the Asian area and from the Ionian area, which would be the most endangered lineage?
Probably the Ionian Sea, because it's blocked. It's blocked by the Quintas mountain range
Okay.
that separates the continental Greece in two parts. Probably the Ionian.
See, Ricardo.
Then another question was, because a lot of these species are very similar genetically and morphologically and so on, and because you only use one genetic marker to look at what they were, you could obviously identify them through the pores and stuff. I was just wondering whether anyone has ever looked into hybridization between these species.
No, the only studies that I have seen dealing with genetics were they only used molecular markers, mitochondrial or nuclear.
But not the two at the same time.
No.
Okay.
And now there is a debate, and maybe this group belongs to Gobiidae or not. They are not Gobiidae. That they cluster with some lineages from the Indo-Pacific region.
So they split Gobiidae family? They divided Gobiidae family and
No, now
Gobiidae is a
the last 10 years, Gobiidae is considered a family, but one American girl studied the gobies in the family level and saw that the Christos clusters with the sister line from the Gobiidae. So there is a debate
on that.
on that.
See, Mike,
No,
you were going to ask something?
was it also the people from Egypt, they published a paper on it. It was a marine species, but where they identified a biogeographic barrier around there, the agency, I think it had to do with large currents.
I know some people from Israel that work with behavior,
But
with
No,
sound
no,
production.
No, this was with phylogeography of Christos, and they looked at all these barriers within the Mediterranean, and one of them,
was the
I
Ionian,
think, was that
between the Ionian and the Asian Sea.
yes, but and those species were
Marine
separate.
gobies.
It wasn't for gobies,
Mhm.
it was for homocentrics, Christos. And yeah, so that barrier, I understand how it can separate everything, and it kind of makes sense with all things that have been described so far. But then here there are populations that are so close together, and yet, or species that are really right next to each other, and yet they're speciated, and there is no dispersal, or there's no connection between them.
In the freshwater fishes in the Ionian side, also the Yugoslavia area, the Adriatic area, the Balkans. It's amazing the pattern of endemicity. A lot of the species and species richness, a lot of the species are a very restricted area in freshwater. It's really
That makes sense because the larvae,
really
the larvae can probably not stand being in salt water, so it's a big barrier to go between one lake to the other. But there were some species, nonetheless, that could live in both freshwater and marine.
See, but even in sticklebacks, the lineage of the Adriatic is completely independent from the European.
And they are as close together as these.
That I could not know. Started to read it at the beginning of the week, so I haven't read that. But if there were different markers, by far, where there were
With different markers, you could eventually look at. That was the other question. Could look at how separate these species really are or not. Because it would be pretty straightforward to know if the mother was one species and the father was a species, then the mitochondrial nuclear should know.
Sim, with the nuclear marker, yeah. Mais alguma pergunta? Então, pronto, ok. Thank you, Christos, for your presentation.
vez e até às próximas Quintas do Mar, que serão em 2015.
Quintas do Mar é.
Do Mar é. E ainda não sabemos exatamente qual será o molde e os convidados, mas saberão ainda.